Good morning, everyone. My name is Hira, and I am currently pursuing my master's in biostatistics. And I was a CFAR intern during the summer, and I joined Tamara's lab, and I still continue to work with her and Dr. Cliburn Chan. So my topic is investigating the impact of pre-existing CMV immunity on the risk of HIV acquisition in HVTN 505 trial. So the general outline of my oral presentation would be to go through the introduction and how we formulated and we came up on this question, what was the data set that we used, and a summary of the results that were generated during the internship itself. And then there'll be a couple of conclusions and acknowledgements. So just to give you a brief overview of HVTN 505 efficacy trial was that it is a vaccine regime designed to elicit HIV-specific multifunctional T-cell responses and abstinence of major circulating strains. There are basically three things that you should remember here, that there are a total of 4 boosts for this vaccine. The first 3 were at week 0, 4, and 8, which were of DNA prime, and then are at 5 boost at week 24. The population, the general population was 2,504 circumcised HIV negative, AD5 negative men or transgender persons who have sex with men, ages 18 to 50. The overall results of this vaccination were that there was no reduction of HIV-1 acquisition or viral load set point. But again, in order to make a better vaccine, the data was analyzed from the perspective of how the different immune functional measures are being correlated or are correlated with the risk of HIV acquisition, the presence of this vaccine or treatment. So the correlates analysis done earlier to this study were the transmitted virus and showed a vaccine-induced sieve effect. And then there were a couple of immune functions that were shown to be correlated, highly correlated with the risk of HIV acquisition, such as CD8 T cells. And then in the presence of, if the CD8 T cell polyfunctionality was low, the N-specific IgG, they were also shown to be correlated with the risk of HIV acquisition. And lastly, the N-specific IgG engagement of Fc gamma R2A mediated ADCP by monocytes and IgG3 binding breadth inversely correlated with HIV-1 acquisition and viral load set point. So now, just to give you an overview of CMV. So CMV is a chronic infection with periods of latency and reactivation, and it works through DNA virus with tropism for myeloid epithelial fibroblast and endothelial cells, and it can be cultured via different ways. The clinical features are, there's a broad spectrum of clinical features associated with CMV. It can be asymptomatic to considerable morbidity and mortality. The prevalence is high, and it goes on higher as the age group changes, and it has been linked to immunosenescence and accelerated immune aging. So why did we land on this question that why do we need to investigate that CMV can have a potential impact on the working of this particular vaccine? So in the past, this study is basically a flu vaccination study where it has shown that CMV actually enhances the vaccination responses in young people or in a certain cohort. But in another study, which was an Ebola vaccine study, it has an opposite effect. So having CMV actually renders the vaccine less, working less. So that kind of pushed us towards investigating that why we need to see the impact of CMV on our vaccination, on HVTN 505 vaccination. And one thing to remember was that since the design of our vaccine was more close to the Ebola vaccine itself, so we hypothesized that it should have a negative effect, that having CMV will actually have a negative effect and it will make you more prone to acquire HIV. So then we landed on the study questions and we formulated these study questions. So the first one was an exploratory analysis. So does the pre-existing CMV immunity modify the relative risk of HIV-1 acquisition in a vaccine efficacy trial? This does not contain any of the immune features that are present in our data set. Then which baseline and post-vaccine immunological features are modulated by CMV infection? And lastly, is there a CMV-associated immune signature that modifies the risk of HIV-1 acquisition in the vaccine efficacy trial that we're looking at? So in order to answer these questions, the data set that we used was 177 functional measures for 44 cases and 145 controls. And some of the immune features that were present in our data set, they include magnitude and breadth of vaccine-specific and CMV-specific T-cell responses, ADCP scores, the magnitude and breadth of HIV-specific IgA, IgG, and IgG3. Then the last two ones are the ones that were recently collected during the summer in Tomorrow's lab, and we added it to the mix of the data set that we received. So it was the systemic cytokine or chemokine measurements that were received pre and post vaccination, and magnitude of CMV-specific IgG responses, which was pre-vaccination. All of the other where it is not specified that when they were gotten, they are post-vaccination functional immune measures. So the first one, coming to the exploratory analysis, which was the CMV infection, is it associated with the risk of HIV-1 acquisition? We looked at simple odds ratios and the risk ratios, and we found out that the risks and odds are actually higher in CMV positive people as compared to the CMV negative people. And similarly, when we adjusted for a couple of confounders, which were the demographic confounders, and the treatment or the vaccine itself, we found out again the same relationship where CMV positive status people compared to the CMV negative people had higher odds or they were at higher risk of acquiring HIV. So this was another analysis that we did. So this was a part of where we wanted to see, okay, which immune features or which immune functional features are associated with CMV. We performed a multivariable analysis, ran a couple of linear regressions, and set a Q-value threshold of 0.2 and found out that these variables or these subgroups are actually associated with CMV infection itself. This includes baseline systemic cytokine levels, Fc receptor binding, magnitude and breadth of HIV-specific IgG. There was a delta value of systemic cytokine levels as well, which is basically a change in post-vaccination and pre-vaccination, magnitude and breadth of vaccine vector-specific CD8 T-cell responses, and magnitude and breadth of HIV and specific CD8 T-cell responses and ADCP scores as well. You can see that the one in negative, they are showing a protective effect, and the ones in positive are having an opposite effect. So these are a couple of data points that we can see. These are a couple of box plots that we plotted. Here are the CMV negative population and CMV positive population, and we can see that there is a protective effect in the ones where the uninfected population is a little higher. So these are the baseline systemic cytokines that are shown to be associated. Then there are delta systemic cytokine values, which are post-vaccination minus pre-vaccination. And then there are a couple of cellular responses and humoral responses that are shown to be so significantly associated with the CMV status. Now coming to the bigger question that how CMV status is associated with the risk of HIV acquisition. For this study, we actually handpicked and shortlisted a couple of features or immune measures that are either, so the pool of variables that were chosen for this particular analysis was all those features who have either been previously linked, as mentioned in the correlates or the previous studies, who have previously been linked to the risk of HIV acquisition. And in addition to those, all the new data that we've generated was added into the pool of this larger feature set that was inputted into this Lasso logistic regression model. And we wanted to see that while we are controlling for all these features that have been shown to previously been significantly associated with the risk of HIV acquisition, how they are related, how CMV status is it still associated with the risk of HIV acquisition. And it showed that CMV status is actually associated with the risk of HIV acquisition when we control for all these additional features. The ones highlighted in yellow here are basically also associated with independently associated with CMV status as well. So we showed that CMV status is actually linked with the risk of HIV acquisition. And then here, there are a couple of data points. Again, now the box plots are uninfected versus infected people who down the line in the vaccine efficacy trial got HIV or did not. So these are the baseline systemic cytokine values that are shown to be associated with the risk of HIV acquisition. And then there are delta systemic cytokines, a breadth of HIV-specific IgG. And then there are a couple of T-cell responses, magnitude and breadth. Here, one thing to notice here is that this one here shows a protective effect. So the uninfected population, if it is higher, it is showing a protective effect. The conclusions were that what we did was employ a systems vaccinology approach, and we found that pre-existing CMV immunity is associated with increased risk of HIV-1 acquisition. The baseline elevation of systemic pro-inflammatory cytokines, depressed mitogenic and chemoattractant cytokine responses, and diminished protective HIV-specific cellular and humoral immunity and antibody functionality. And our findings suggest that a possible role of pre-vaccination interferon gamma pathway activity in modulating HIV-1 acquisition. In the end, I would like to acknowledge Dr. Cesar Lopez. He was my mentor, and he has been a tremendous inspiration for me. Dr. Tomaris herself, she has been super helpful. Dr. Janice McCarthy, who is a part of biostatistics department and who has been my quantitative mentor through CFAR internship. It has been wonderful. And I would like to thank all the members of the Tomaris Lab, Lindsay, who helped Cesar to acquire data, Yuji Fong, who has been working on HVTN 505 trial for a while now, and Mohammed Faizyab Ali, who is part of University of Iowa. And we've been having a couple of discussions. So I would like to thank CFAR for the wonderful opportunity, and I continue to work with Dr. Tomaris and Cliburn Chan in the future. Thank you.
Great, thank you so much, Hira. That's a fantastic presentation. So this abstract is now open for discussion. We have a few minutes to take questions, and I may start while folks are typing into the chat box. So this is really, really interesting data, and I have a couple of thoughts, but maybe one question, thinking about the impact of prior CMV immunity on responsiveness to vaccines, something that has been described with other vaccines. A question that I would ask here is, does that mean that CMV immunity, which is obviously quite common as you mentioned, should be considered in clinical trial design for vaccine programs? Is it something that should be controlled for in some way in the design, whether it's stratification, et cetera, given the potential impact on the outcome of the trial?
I think that was the hope, that this is one of the potential things that we have to look at when we are considering vaccine design. So yes, I think to answer your question, yes. I guess CMV immunity should be considered when designing a vaccine for HIV.
And then one other follow-up question to that is, this is really interesting data that you're identifying in terms of the pre-vaccination pathways. Do you think that there's potential as you all do more work and try to understand and kind of really identify particular deficits that may result in poor responsiveness of strategic adjuvants, for example, for vaccines to boost those exhausted pathways?
I think in terms of, are you asking about the future directions for the vaccine design itself?
That's right, given the CMV impact in terms of responsiveness.
I think the interesting findings of this study were that we have CMV is significantly associated with the risk of HIV acquisition. And in addition to that, we looked at some baseline cytokine values that were associated. Obviously, this will give us a direction that these are the possible avenues that we have to consider when we are answering the bigger question of vaccine design.
Fantastic. All right, so I don't see any additional questions in the chat box. So thank you so much, Hira. Really great work.